Yes, I started.
Okay, fine. Okay. Hello, everyone. So first of all, I would like to thank Alex and Christoph for the kind introduction and also for inviting me to speak about plant-pollinator interactions. And today I'm going to talk about how flowering plants, and in particular their chemistry, their nectar chemistry, can affect pollinator behavior and foraging. As you know, probably, I mean, has been a very long-lasting coevolution between angiosperm and flowering plants and pollinators. And this very long-lasting coevolution has made both plant and pollinator reciprocally dependent one another. And the interaction between plants and pollinator mostly rest on a mutualistic exchange. So in this coevolution, basically, flowering plants offer flowers that dispense rewards like pollen and nectar, and in turn, pollinator vector their pollen to a specific flower. Of course, the story is more complicated than that. So the exchange is not just mutualistic, and sometimes we have cheating, we have exploitation between the two parties. I guess the story between plants and pollinators can be seen from both sides, the pollinator perspective and the plant perspective. So from the pollinator perspective, of course, a meadow, so especially the natural one, so a good meadow is a really rich marketplace consisting of a lot of different flower species that most of them offer rewards. And the fact that, especially during blooming season, you have out there a lot of different plant species and flowers, basically doesn't make necessarily the foraging task an easy task. And foraging, as Elodie Bieder, for instance, in the previous seminar showed very well, foraging can be actually quite complicated, especially if you want to make economic decisions in order to ensure a net gain of resources. So when you have a lot of different species of flowers that differ in shapes, in color, in fragrance, and most important on the rewards they provide, it can be really difficult to make optimal decisions. And of course, pollinators, and I'm not just talking about bees and bumblebees, but of course also mammals and birds, naturally excel in their cognitive abilities, in particular navigation in the environment, learning and memory, but also the reward evaluation or flower discrimination. And when pollinator make decisions during foraging, they mostly rely both on innate and experience-dependent preference. Everyone that is working with bees or bumblebees, for instance, is aware that bumblebees, for instance, have this innate predilection for blue or purple flowers, and that's because they have this kind of innate preference, this predisposition for approaching this type of flowers. But they also are very good, and they exhibit rapid sensory learning, so they can really update the information. They can approach different flowers, even unpreferred one from the color point of view. And as soon as they realize that those flowers actually provide a better reward, they can really start foraging only on these flowers. And once they find a very profitable food source, they often show this solid flower constancy. So in this process, basically, they stick on this type of flowers and they ignore other flowers that are in the surroundings, even equally rewarding flowers. And one aspect that makes foraging really, really challenging at times is the unpredictable ecological circumstances. And so, of course, flowers and blooming, are ephemeral. So they change really in space, in time, very, very fast. And these actually, even, the reward they provide can really change over days, but even, within the same day, depending on temperature, depending on the presence of other pollinators, for instance. And so pollinator, not only must be able to find, recognize, and memorize the best flowers, but they also need to readily react to these changing situations. So they need to be really flexible, and cognitive plasticity is therefore really key for the foraging success. And from the plant perspective, which is the other face of the coin of the story, pollinators are clearly a unique resource of Grady customers that they must attract first and then retain. And for any flowering plant species, attracting and retaining pollinator, the pollinator that they actually want, it's really a top priority. And for doing these, plants usually, of course, evolve with flowers. And flowers have a variety of cues and signals, like the color, like the shapes of the petals, and that actually allow them to make them really, visible, and so these help them to actually attract pollinators, specific pollinators. And attracting pollinator is just half of the story, because once attracted, of course, they need to build the loyalty of pollinators, and they can do this providing rewards. And rewards can be, of course, nectar and pollen. And nectar in particular, it's very important because it's the fuel, it's the energy of most pollinating species. And pollen also, which is clearly important because it's the object that has to be vectored to specific flowers, can be also act as a reward. And floral nectar clearly is very important because and it's a very complex mixture of chemicals. So you can find carbohydrates, lipids, and of course water, minerals, and vitamins. And all those molecules are have a lot of caloric and let's say value, okay, from the metabolic point of view. But in floral nectars, actually, there are many other molecules that overall we call secondary metabolites. And those secondary metabolites actually are really diverse. So we have several different families of molecules. So we have, for instance, phenolic compounds, we have tannins, terpenes, flavonoids, alkaloids, and non-protein amino acids, among, of course, others. And those molecules, of course, are really diverse in terms of structure. So also the activity and the biological function can be really, really diverse. And as so far as put forward many different hypotheses that try to explain why those molecules are present in nectar. And the first actually hypothesis was the one of the pleiotropy. So the idea that in general most plants have a lot of different compounds. Many are the same that we found in nectar, in very high concentration in the vegetative part, in the green part of plants. And this because at very high concentration can be toxic, can be distasteful to vertebrate, insects, and herbivores in general. So it's a kind of defense strategy that plants has. And so the idea was, and it can be still, I mean, that those molecules just go through the flower and reach the nectar as a byproduct. Of course, this is a bit counterintuitive because, I mean, nectar is something that, even pollen, is something that has to attract pollinators. So the fact that you find these molecules in nectar can be, difficult to explain. But we have to be careful and say that the concentration of those metabolites in nectar is far lower than the one that we find in vegetative parts. Another hypothesis is that those molecules have antimicrobial activity, and so those molecules can actually help the plants to keep their nectar safe, let's say, because microbes and fungi and yeast, for instance, can degrade the nectar itself. And then, I mean, more recently, has been well suggested and demonstrated, I would say, several times that actually at least some of those secondary metabolites can really act as attractants. So their role in nectar is something that it could be very important for plants because they can really improve the the visitation rate by pollinators. And there are, I mean, seminar paper from Baron and Geraldine Wright, Leon, that actually demonstrate that several alkaloids, in particular cocaine or caffeine, can really act as mind-altering chemicals. So those molecules actually serve as a form of floral deception. So they can really seems to manipulate the foraging behavior of pollinators, at least bees and bumblebees. And in this way, the plants manage to increase the quality and the quantity of the pollination itself. So today I first talk about one of my papers that I performed when I was postdoc fellow in Chitka lab. And we at that time studied the effect of nicotine, which is another alkaloid that can be present in nectar of some plant species. And we try to understand what is the impact of caffeine on bumblebee learning and preference over forage. So in this experiment, we take advantage of Bombus terrestris and the fact that we can easily work with free-flying insects in the lab. And we first asked whether actually bumblebees had a preference for nicotine-less nectars, and also whether nicotine can actually influence positively or negatively associative learning in foraging bumblebees. Well, this is actually a picture of my lab, but I essentially reproduced what we use in Chitka lab. So here you see, I don't know if you can see my pointer. Anyway, in the small box, the wooden box, you have the colony, bumblebees. Then you have a second chamber, which is kind of outside of the nest. And then the colony, the box is connected to a flight arena. In this flight arena, you can use artificial perspex flowers. So usually during each day, we let all the bees forage inside the colony, so they familiarize with the perspex neutral flower. And we also marked individually motivated bumblebee foragers. Afterward, we basically set a preference test. So we present bumblebees with two types of flowers that actually differed in color and also in the reward. So one type contained nicotine in sucrose solution, and the other type just sucrose solution at the same concentration. And we test three different groups of bumblebees using 3 different doses of nicotine. 2, the high and the low, were within the natural range, so the range that, I mean, the concentration that I mean has been demonstrated in nectar, and 1 was really above the natural range. And we let the bees forage for 100 consecutive choices to see actually if bumblebees preferred or not the flower dispensing sucrose solution plastic. And what we found was a dose-dependent effect. In particular, the very high dose, the 50 ppm, in a way deterred a bit the bumblebees foraging, and so foraging bumblebees basically prefer the flowers dispensing only sucrose. But this deterrence disappeared or even turned into attraction when the nicotine was present at lower concentration. And so this suggests that bumblebees can probably perceive nicotine in nectar, or that nicotine can actually directly acting on the foraging in the season and the wanting actually. In the second experiment, then we test the effect of nicotine on learning and memory. In this case, we set up a test consisting of forward learning. So in the first phase of the experiment, each market bee could enter in the arena and could find 2 different type of flower. One type of flowers, for instance, the blue, okay, were unrewarded, so only dispensed the water, while the purple was rewarded. And in one group of bees, the rewarding was just plain sucrose solution. And in the experimental bees, we added nicotine to sucrose. And again, we tested 3 different concentrations of nicotine within the natural range and 1 above. And we let the bees one at a time, of course, make 50 visits. And 50 visits, it's enough for the bees to basically understand that one type of flower is rewarding and 1 is not. So they stop visiting the unrewarding one. After finished this phase, we basically swap the content of the flower. So in this reversal learning task, for instance, the blue flowers that before were unrewarded now were rewarded with sucrose solution, in both cases in control and experimental bees, and the purple that before were rewarded now are unrewarded. So at the very beginning, of course, bees visit the wrong flowers, and then they need to update the information in order to improve foraging. And what we found was that overall the bumblebees of the 4 groups improved the performance over the 50 visits in the forward learning phase, and nicotine improved accuracy of these. And again, the effect was dose dependent. So the natural concentration of nicotine enhanced the learning, the rate of learning, so the speed of learning, while the other 2 doses actually did not. But the interesting part was even more the second part of the experiment. So in the reversal learning, again we found that bees were more or less able to update the information, but again bees exposed to nicotine in the forward learning stay more faithful to the previously rewarding flower. And the nicotine at the very high and natural high dose had the strongest detrimental effect. And you see that actually most of them, I mean, failed to relearn, to update their information. And here in this plot, you see that there is a lot of variation in the bumblebees performance, but for instance with nicotine with 50 ppm, we have, we had actually several bees that never managed to swarm. And this might be consistent to, I don't want to say addiction-like, because of course, addiction is something that it's difficult, to demonstrate, but something that goes in that direction, because actually they pay a cost because of their previous experience with nicotine in the forward learning phase. Okay, so alkaloid is represent a family, a type of molecules that are quite often found in nectars. But other classes of secondary metabolites are far more common actually and abundant in nectars. In particular, NPAAS, that are non-protein amino acids, so non-essential amino acids, are probably the most ubiquitous and abundant class of nectar secondary metabolites. And interestingly, some of them, like for instance GABA and beta-alanine, are also neurotransmitters or neuromodulators. And GABA and beta-alanine in particular are the most represented in the few nectars that have been studied for this. And interestingly, in at least from one paper in South America, it seems that NPAAs are most frequently found in hymenopteran pollinated species. GABA, in particular, also is the most important inhibitory neurotransmitters in insects, and not only in insects. And its function, we know, being essential for olfactory processing. So, because in the literature there were really few papers, on the topic, we decided to investigate the effect of those molecules, of those non-amino acids on several aspects of honeybee behavior and learning and memory. And this work have been performed in my lab in Florence, mostly by Daniele Carlesso and Stefania Baracchi, and that I thank here, but also Elisa Paspini and Giacomo. At that time, they were all students. Actually, now Daniele and Stefania are both PhD. And so in this study, we basically studied the whether, beta-alanine, GABA, ornithine, citrulline, and taurine interact with gustation, feeding preference, learning and memory in Apis mellifera. In this case, we wanted to test several molecules and several aspects, from gustation to feeding, learning and memory. And for these reasons, we prefer to use Apis mellifera because we take advantage of a very common protocol, which is the Proboscis extension response. That it's very, evident in honeybees. So in this protocol, basically, the bees are harnessed, so they are completely blocked into these tubes, and they can only freely move antenna and mouthpart. And we know that they exhibit an innate response, a reflexive response. So, sorry, I guess I have to quit from this. I don't know if you can see. When you basically touch the antenna with sucrose, they start leaking the sucrose solution. So this is the proboscis extension response. So this is actually a reflexive response, and as such, it can be conditional. And there are many different protocols that we can use. And this technique, I know it's really far from ecological circumstances. So they are tethered, so they cannot move. But it has the advantage that you can really standardize for many different aspects, in particular appetitive motivation, which is a very important point because you are studying actually appetitive, well, in this case, olfactory learning. So in this protocol, I guess that many of you probably are familiar, but I will explain it very fast. So before conditioning, so before starting with the training, if you touch the antenna, the bee exhibit this proboscis extension response. And if you release an odor, which is at this time neutral, nothing happens because this odor has no balance, no value for the bee. During conditioning, you present the bee first with an odor, and then after a few seconds with a sucrose stimulation for 4 or 5 seconds. Sorry. And in doing this, basically, the bee starts understanding that the odor predicts the arrival of the reward. And so they start responding only to the odor, which is now conditioned, and it triggered the proboscis extension response without the presence of any sucrose. So over time, you see in this plot, you have several conditioning training, and see that the proportion of bees that learn increase over time. And then you can test the memory recall, for instance, 1 hour or 2 hours later, or 1 day, or even 3 days later. So you can really test different types of memories. So in the first experiment, we basically try to understand whether bees can or not detect and discriminate with their antenna these 5 non-protein amino acids. So in this protocol, which is something different from what I've shown just right now with odors, it's a chemotactile conditioning, and the CS plus is represented by a stimulation on the antenna with water plus 1 of the 5 amino acids. So after this stimulation with water and amino acids, we provide a reward, and then we present also pure water without amino acids, and we provide a punishment salt. So during the conditioning, the insect basically, the bee learn to respond to the CS plus, that's it, that is the amino acids, but not to pour water because it's punished. And so learning can occur only if bees can actually detect the difference between the CS plus and the CS minus, which is the amino acid. And what we found basically is that bees were unable to detect those 5 amino acids with their antennas. So they overall increased the responsiveness to both stimuli, but they never managed to respond to the amino acid and not to water or the other way around. In the second step, we then asked whether actually honeybees showed a preference for those molecules. And this is because also, I mean, honeybees can also detect tastans by using mouthpart or tarsi. And so in this experiment, we blocked our free walking bees into this 15 ml tube, and then we provide the bees with 2 capillaries. One was filled with plain sucrose, the other one with plain sucrose plus the amino acid. And we monitored the volume consumed within 2 minutes. And what we found here was that overall there was no preference for any of the 5 tested amino acids. So here you have a delta consumption between the 2 capillaries. So on the 0, it means that they have no preference at all. But overall, we did not find any preference, any evident preference for those ones. Preference clearly can, arise over time. And this, especially if after consuming those molecules, for instance, you have effects like malaise or phagostimulation. For these reasons, we prepare a group of 10 bees in cages, and we allocate them to 3 different conditions. So in the first condition, the bee could, have access only to sucrose. In the second condition, they have access only to amino acids in sucrose, of course. And in the third condition, they could choose between one syringe with sucrose and the other one with sucrose plus amino acids. And what we found was that bees, again, did not consume more or less amino acid solution than control. So over a longer period of time, there's no any change in preference. And this is consistent with the fact also that actually we did not observe any mortality rate difference under different feeding regime. So in a final experiment, actually we tried to understand whether Proboscis, whether those molecules could actually affect, improve, or block appetitive learning and memory. For doing this, we set up two different set of experiments. In a fourth experiment, we basically provide the bees with sucrose plus the amino acid during the conditioning. In a second set of experiments, we basically provide we provide the bees with the amino acids 2 hours before starting the conditioning test. What we found in this case was that at least 2 amino acids, so beta-alanine and GABA, actually affect learning and memory. This when the 2 amino acids were provided during conditioning. And in particular, beta-alanine and GABA, so both basically improve the memory performance of bees. So the learning rate was faster, and at the fourth trial, the proportion of bees that actually respond to the conditioned odor, the CS+, was significantly higher. And this also translates in a better memory at 2 hours later. In particular, beta-alanine, improved the response to the CS+, but not to the nod. The nod is a novel odor that we present during the test to see whether there is also a generalization, so an overall increase in responsiveness. And specific memory was not affected. Specific memory is the proportion of bees that actually responded to the CS+, so the conditioned odor, but not to the novel odor. In case of GABA, the effect was more evident for the specific memory and was a trend, but not significant for the CS+. For the other 3 molecules, so taurine, ornithine, and citrulline, we found basically no effect. So neither, the acquisition nor the memory was affected by those molecules. And this suggests that different molecules belonging to the same family actually can have different effects. In the last experiment, where we first, fed the bees with this amino acid, and 2 hours later we start with the conditioning. In the conditioning, the US was just plain sucrose. We found again an effect, but this time the effect was at the opposite. So in this case, for instance, beta-alanine and taurine both impact negatively the acquisition performance, so the learning, and so the learning rate and the speed actually was lower. And well, surprising, then we still found a positive effect on the response to the CS+ in one case, in the case of beta-alanine, and enhanced specific memory for taurine. And this is, I have to say, well, quite tricky difficult to explain. Similar result was also found for citrulline and GABA. The only difference in this case was that actually GABA also impaired the memory recall. So again, here each molecule seems to have its own specific action, and ornithine actually had no effect in any aspect. And so to conclude this part, and honeybees seems not to be able to detect those molecules with their antenna, and they also do not prefer nor avoid those molecules, either because they cannot taste them with the mouthpart, or because probably they can taste with mouthpart, but they actually do not prefer or some amino acids like GABA in particular and beta-alanine can be psychoactive and can act positively on learning and memory in simple olfactory learning. But depending on the time when they are consumed, basically, they can also impair learning. And we think that in this case, probably those molecules could possibly interfere with the hunger signal, reducing the appetitive motivation and thus learning. But of course, this is in part speculative because I mean it's really difficult to find a real explanation. And then now I will be really briefly. I promise this. So this is something that is still ongoing. So is something that we are currently working at, and we are looking more closely on the GABA effect, and we move on bumblebees again, and we wanted to test the effect of GABA in this, let's say, setup that it's a little bit more close to an ecological approach. At least, I mean, they are not harnessed, they are actually active foraging in this artificial scenario. And the protocol was exactly the same of the one that I showed you for nicotine. So we tested for the preference, and what we found again is that there is a huge variability. We tested 3 different concentrations of GABA, 2 within the natural range, 1 higher. But in all the 3 cases, bumblebees basically ignore GABA, so they had no preference for, I mean, flowers with or without GABA in nectar. And then we test again the effect of GABA on forward and reversal learning phase. And in this case, what we found was that well, the high dose had no effect, but for the doses within the natural range, there was a mild but still significant effect on the reversal learning. And forward learning was not affected, reversal learning was, and indeed these, with these natural doses, were more fightful, a bit more fightful to unreward flowers. So this suggests that GABA might be an active role in increased fidelity to flowers. And the last experiment, and you have to keep in mind that in this experiment we were testing actually flowers of different colors. So we are in the visual domain, in the visual level. So in an additional experiment, we use actually artificial flowers that were all identical from the visual point of view, and we provide half of the flowers with one odor and second half of the odor flowers with a different odor. So we tested the effect of GABA on olfactory learning. And again, we did not find any effect on the forward learning, but we found an effect on the reversal learning. But in this case, actually, the effect was the other way around of the visual modality. So in this case, GABA were less helpful to unrewarded flowers. So in this case, GABA seems to facilitate the reversal phase, and this clearly it's difficult to explain from the ecological perspective, because in this way GABA might help bees to be more flexible. Nonetheless, it's probably more easily explainable in terms of pharmacology, because we know, for instance, that in harness bees, I mean, if you block the GABA receptor, basically you block reversal learning. So it makes olfactory learning in harnessed honeybees. So this goes in the same direction of our research on olfactory learning in bumblebees. And I guess, yes, that was the last slide. So I've done. So thank you for your attention. And again, thank you to Alex and Christoph for inviting me. So if you have questions, please feel free to ask.
Yeah, thank you, David, for an interesting talk. So, okay, there's one hand. No, it's clapping. It's not raised hand. Are there any questions? Okay, then, David, then I'm asking.
Yes, yeah, sure.
So, I actually was wondering whether you will have at the end a summary of how you think these plant-produced substances affect the foraging behavior, right? I mean, how to say, so kind of like listening to your talk, you know, it was once about visual learning, then about olfactory learning, then you have the different substances. And I was wondering kind of like whether there could be like a larger framework, then kind of like how to say, if you would demonstrate that some of these substances affect the learning, right? And you hypothesize that that has something to do with the evolution of plant products, then you might have to see it kind of like a variety of these products in different plant species or something like that, right? That to then demonstrate that there's an evolution or something like an adaptation.
Yeah, I guess I mean, one of the next steps is, of course, make more studies on different molecules and try to, for instance, just see whether those molecules can be framed in a kind of flora syndrome, for instance. So it would be very nice to compare nectars from different species of plant, for instance, that are co-evolved with, for instance, not non-generalist pollinators, to see actually if those molecules can, I mean, if yes, these molecules can be co-evolved with the specific pollinators. Of course, I mean, many of those molecules are molecules that you can really find in any pollinating animals, because GABA, for instance, is the inhibitory transmitters in vertebrates and vertebrates as well. And caffeine, cocaine, or nicotine are the agonists of the acetylcholine receptor. So yeah.
Okay. Rajbir, do you want to read the question in the chat box?
Sure. So the question says, Dr. Baracchi, thanks for your talk. Yeah.
There's some background noise.
True, I'm in a breakout room, so yeah.
Okay, good.
Okay, I can check the chat by myself.
Okay.
Thank you for your terms. So was wondering if you have thought an approach to find at the neurobiological level a technique that might mirror your findings. The results are really exciting and maybe important also to advance neuroscience. Yes, that's a very good point. Yes, probably this is feasible, at least with tethered bees. So we might have a look at the effect of those molecules under, for instance, functional imaging, and see how the presence of those molecules and, for instance, an odor can be coded by different glomeruli or, I mean, neuronal activity at the level of the mushroom bodies or any other part. So this probably is one of the best way. A different approach might be a more pharmacological approach. So you can, for instance, provide bees or, I mean, any type of pollinator with those molecules in a foraging or learning process, and then try to measure biogenic amines in the bee brain. And biogenic amines might be involved because they are neuromodulators, and octopamine is involved in appetitive learning. And we also have now evidence from this very, very recent publication that also dopamine, like in mammals, also are involved in the motivation of pollinators, of bees. So this might be a different but valuable approach.
Okay, I don't see another question. Then I'm asking another one.
Yeah,
so last week, I mean, Felicity Mood kind of reported about experiments with, okay, I have problem pronouncing it, neonicotines. I mean, with imidacloprid, I mean,
yeah,
right?
Yeah.
So, okay, similar to nicotine. Yeah. And the interesting thing was that she demonstrated that there was a different effect on olfactory learning and visual learning. And we had some discussions about that. And I think you also showed, and you use different substances, but how to say, so people use visual learning and olfactory learning, then testing for learning, but then actually, because also, as you mentioned, it's different modalities. And kind of, so Felicity showed a strong effect on the visual system, way stronger than on the olfactory system. Is that, and you did your nicotine experiments only with a visual learning?
Yeah.
Did you know about experiments with nicotine and PR? And is there also kind of like a difference on that effect?
Sorry, I missed the last part of the question.
Is there a difference of the effect of nicotine on visual compared to olfactory learning? That's basically the question.
Yeah, we actually, at least with nicotine, I mean, as far as I know, there's not a single paper addressing the effect of nicotine on the two modalities. So there's no a real comparison. Of course, nicotine, I mean, is expected to have a stronger effect on the olfactory modality because you have a lot of nicotinic receptor in the antennal lobes and the tract that project neurons to the mushroom body.
But no, it was actually the opposite. It was a stronger effect on the visual system than the olfactory system. And then we were kind of discussing whether there are more acetylcholine receptors in the visual system. Maybe Albrecht Hase can add. I mean, he is kind of, I mean, I think he knows more about that, and he is in the audience.
Yeah, so I don't know actually.
Yeah.
But I mean, another aspect that we should consider is the concentration. And because of course we have different types of receptors, and the different types of receptor could be more or less sensitive to different concentrations. So it might be that the same substances have opposite effect or act on different parts of the brain depending on the specific type of receptor. Yeah.
Okay, and that's then my last question. Then. So there is a preference of testing these substances for learning and memory, right? Because, okay, it's about visiting the same flower or similar flower again. But it could be that all these substances might have kind of more direct effects on the muscles, for example, or on the heart, which then might kind of affect actually the flight behavior. Yeah. And this is difficult to test in the lab assay, but that could be then even kind of more stronger effects, right?
Yeah, that definitely. For instance, GABA, it's very active at the level of the muscle, so it could sustain flights, for instance. So this is something that we want to investigate with flight meals. Because I mean, bees can be easily tethered on flight meal, and you can quantify the flight activity. And, and so the idea is to provide different diets with different molecules and test whether, I mean, flight activity can be boosted by those molecules. Yeah, but I guess, I mean, what we are observing in our study, my studies, but also studies of caffeine, is a change in probably motivation and learning and memory per se, not just flight ability or flight speed. It's the motivation that changes.
Yeah. Okay, so there's a question. There's a hand raised by Tapovan Farms. Please, kind of unmute yourself and ask the question. You can also show your video. I mean, your face.
Yeah, David. This is probably not directly related to what you are doing, but have there been any experiments to change the hostility of the bees by lacing it with some chemicals in its feed? Like for Dorsetta, can we pacify the colony before examination by adding some substance in a syrup and feed it? Or can we basically make a colony wake up early in the morning by giving it some coffee kind of substance? So has there been any related work in this regard?
Okay, I'm not sure I understand the question. The question was about the timing?
It was about basically whether one could use substances to direct the foraging activity. So
yeah. I mean,
giving them caffeine in the morning, and then you get kind of more activity.
Yeah, like how we take coffee in the morning. Yeah, actually, I mean, there are very beautiful paper that very recently showed how I mean, you can prime bees with those molecules and help them to target the species of plants you want. And this is something that it's really can be applied to agriculture. Yeah.
Can you just say the paper or the author?
The paper there was two papers, and one was by Harmon, if I'm not wrong.
Is it Walter Farina?
Farina was the one in current biology. Yes.
Mm-hmm.
There was another one. Yeah, the last author was Walter Farina from Argentina. Yeah.
Okay, so maybe Tapovan Farms can send me an email, and then I'm checking for the paper. No other question so far. Anyone having a question? Let me see that I. No. There was one question, David. So in one of your figures, that was a box plot. It looked like a bimodal distribution, or was I wrong?
Yeah, I can show probably.
Yeah.
Not. Yeah, I mean you are right, and this is probably because in our experiment we try to make sure that the bumblebees actually attach the both capillary with the antenna. And we also, I mean, for half of the bees, we balance left-right. But this bimodal distribution, I mean, might also, suggest that really there's no a real preference. But once they select a capillary, they simply start drinking from that capillary, and they do not change. So that's the reason. But the capillary were really close, so they could alternate basically both.
Yeah.
So I guess this might be the explanation for this bimodal.
Okay. Someone a question? Okay, good then. No, okay, there's no question. Oh, wait a minute, there is one. Let me see. Yeah, Christian Berg. Hello.
Hello. One quick question. You presented data on bumblebees and honeybees. Do you think the kind of foraging they're doing is actually influencing your results? So it could be the findings you find in bumblebees where you have individual foragers be different to honeybees where you actually have communication and interaction. Because we find that diet and neonics affect sucrose sensitivity. So perhaps there's a research that they get actually reset every time they go out or so.
Yeah, so basically it could be that the difference we found in bumblebees and honeybees actually was not simply because of the model species, but because of the way we tested the preference. And so this is of course possible, but in all cases we always test one bee or bumblebees at a time, so independently from the other. So it should be we never test, I mean, foraging behavior in honeybees, and when we test the preference or learning and memory in bumblebees, we always did using one bee at a time.
I think that, Christian, do you want to respond? Then otherwise, I think kind of Christian's question was quite great, because at the beginning you were kind of referring to flower constancy. And actually, I don't know which species, bumblebees or honeybees, are more flower constant and whether there have been comparative studies. So if you, for example, kind of like with this ecological background, right? I mean, I think it would be great, for example, if one would have a quantitative measure of flower constancy, and then how to say then you could decide for a species, you could avoid a ceiling effect, maybe kind of like honeybees are flower constant, whereas others kind of like vary, and then you would prefer those. Maybe you
this is a very, very good point. But I mean, that's why we use, in a way, reversal learning, because in the reversal learning, you can, in a way, test the constancy, So how long you keep visiting the flowers, even if they do not provide any reward. But I mean, the question is very good. So I didn't catch it at the beginning. So the point is very good.
Yeah. So for example, kind of now here, Mario Sol describes honeybees are more constant than bumblebees in nature, and then of course, then one would get into kind of totally complication if you would say then you might have to test scouts against recruits in honeybees, right? And maybe the scouts are kind of like responding totally differently, assuming also that they are on a different motivational state. Yeah.
Yeah, definitely.
So that is interesting. Yeah. And okay, and then I have another comment. It was regarding kind of like the question with which sensilla, if kind of like bees might perceive these substances, and you were kind of going from the antennae to the mouth parts.
To mouth parts,
And I'm sorry, this is because we found it, that I have to say it. But that, in a collaboration with Daiichi Tanimura, we worked on Drosophila, and we worked on a behavior that's called sugar illicit search behavior. And what we found was that sugar receptors in the pharynx are important for that behavior. Yeah, it's not the crop, it's not the mouse part, it's not the antennae, it's in the pharynx. I mean, one can study this in Drosophila easily, right?
Yeah,
but how to say, I think that is, but I was so astonished by that. And I think this is a pathway that is not really understood in honeybees. And,
absolutely.
And particularly, it could be even kind of like a stronger pathway than the sugar receptors. I mean, it might be a sugar receptor. I don't know.
Yeah.
I don't remember what it was, but the sugar receptors on the antennae as well as the mouse parts, because it already indicates that they're ingesting, right? On the antennae, they don't do ingesting, right? So and it is the reward of the ingested food. Yeah. Yeah. So
I guess we need more electrophysiological experiment.
Yeah.
This is the only way.
Morphology or old morphology studies checking kind of like what tensilla are there, right? Yeah.
Yeah. Also because, I mean, probably it could be that some, I mean, for instance, the pollinator can indirectly perceive the presence of some molecules because those interfere with the sucrose sensitivity. So this is still, I mean, controversial. There is a strong discussion on these topics about the fact that, for instance, honeybees or bumblebees can have this receptor for a bitter taste. It could be that the molecule just actually inhibits the receptor for sucrose, so they do not perceive the molecule as bitter but less sweet. This is something that is still, I guess, ongoing, and we need more electrophysiological studies on this point.
Okay, so there's something coming up in the chat. So it kind of like I just repeated. So honeybees are more honeybee is more constant than bumblebees in nature. Then Tapovan Farms, which is from India, comments on Marisol might be referring to mellifera. Can't say the same with Serrana indica, can we? Okay, it's a question. So that's the whole topic of Asian honeybees and whether they are different. And particularly one can say maybe one thing which is important is that if we study Apis mellifera, we really, let's put it from an Asian standpoint. If we are studying Apis mellifera, we have to realize it is not a species, it's two populations. It's Apis mellifera carnica and ligustica with mid-Europeans, temperate populations, right? So, and then Apis cerana, kind of like in India, would be kind of like the tropical population. There's kind of like temperate populations in Apis cerana. But how to say, so temperate and tropical honeybees have totally different nutritional needs because the temperate, they have to collect a lot of nectar for surviving the winter, right? So they have way larger stores. Okay, that kind of like I'm stopping here. Let me see what's, okay, there's a question. How do bees, regulate floral constancy? Kind of probably meaning kind of how is floral constancy achieved, right?
Yeah, probably there are many factors. It's well, for sure. I mean, motivation, I guess, it's something that might affect this flora constancy. And indeed, they start exploring other sources when the flowers, for instance, do not meet the expectation, probably. Or yeah, and this might be and also probably the ecological scenarios, because if you have many options, probably you are more prone to explore other type of flowers. But yeah, I mean, I guess there are probably many different possibilities, but motivation, I would say, probably.
I was also wondering, is there anything known, so how to say, from the plant perspective? Do the pollinator that comes have to have 100% of that pollen of that species, or is kind of like 20%? I mean, so if it has pollen over its kind of body, is kind of like 20% sufficient? I mean, does one know anything about that? I mean, so the question is kind of basically, how many other flowers can you visit and I'm still getting my reward, my pollination done, right?
Yeah.
Okay, Christian says depends on the plant.
Okay.
It's kind of like, yeah, probably we should kind of keep it at that level. I mean, otherwise we, yeah.
The big problem is you have to take the plant now into account as well. So that becomes really, really tricky to make a general conclusion. I mean, it's the same with flower constancy. I mean, it all depends what the alternatives are. And I agree, it's probably the motivation of the colony or the individuals as a whole, if they're constant or not. But as soon as something nicer comes around, I'm pretty sure constancy goes out of the window.
The question could be, for example, whether, so if from the plant perspective, if you're a specialist and compared to a generalist, that maybe a generalist kind of like can deal with kind of like a lower percentage of pollen of the same species when a bee, a pollinator comes, compared to the specialist. I mean, at least one could come up with a hypothesis and then might be testing it or something like that.
It can be. Yeah.
Okay, good. So are there any kind of like other question that I kind of, I think there's no, and here it's already 6:45 p.m. David, I'm thanking you for the nice talk. I mean, we appreciate that you accepted our invitation. I have to tell you what is next week on, and I only can say you it's Nigel Rain from Canada. You know, also Lars Chitka, kind of student or a postdoc, but Nigel hasn't kind of sent us a title. As far as I'm informed, it will not be about honeybees. Then what will it be? Bumblebees again, I think. So we will inform you on the Twitter account, and you will, of course, all get the reminder email with then the title. And we hope to present a poster. So thank you very much, and then good evening. Okay, wait a minute, there's another message. Okay, clapping hands for you, David. So,
yeah.
So, okay, thank you, David. Yeah, and then all of you have a nice day or kind of nice evening.